Hi there, this is AGNR 131 Soil Science, and this is lab exercise 9. It's called Some Soil Chemical Properties. This is part 1, and in your lab handout it is called part 1A and part 1B. So I'm gonna run part 1A and part 1B together, and at the end of that there'll probably be some breaks in here because it's going to take a little while to do that, but we'll show you the whole procedure. So part 1, what we're really trying to do here is number one, we're going to prepare what we call a saturated paste. The goal of this particular exercise is to measure soil salinity. So this is not, there's other ways to do it, but this is the probably one of the better ways to do it, if not the best way to do it. This is how you would do it in a regular research lab. So we're going to kind of mimic that particular procedure. But so what we have to start with on that one is we have to start with some soil, right? So the soil that we want to analyze. Now I've got 4 beakers here. They've all got 4 different kinds of soil in it. And to be perfectly honest, I have no idea what the salinity level of these soils are. They may not even have any salinity. I suspect that some of them probably will. I don't need to use all this soil. I don't even need to use a certain amount. I just am going to put some in. I got a beaker here, and I'm going to take my big one. I'm just going to put some in there. Now, I know I'm going to need a fair amount because what I'm going to eventually do is I'm going to extract this. I'm going to put this through a vacuum pump, and it's going to go in these funnels here, and I'll show you that. So I need to have enough soil to be able to cover the bottom of this particular funnel. So I'm looking at that, and that's probably a minimum. I might need a little bit more, and I can always add some more to it. Now, the other thing I'm going to need here is some distilled water, or deionized water is best. We use distilled because the other one we can't get. And I'm not going to really measure this. At this point, I'm just going to put a bunch in there because what I'm looking for is I'm looking to reach saturation. So it's going to take a little while for me to reach saturation. And so I got a whole slew of torture instruments here. This is one of my favorites. I like this particular one. It works really well. You don't break the glass. I'm always breaking glass. And I'm just going to mix it up. Well, we're not even close to saturation there, so I'm just going to add some more. Now, you really do want to be careful that you don't go too far, but if you do go too far because you're in a hurry, and if you were doing a whole bunch of these, you would be in a hurry, unless they're paying you by the hour in a lab, then you probably wouldn't care. But just go ahead and add some more in through there, and I'm going to show you what this looks like when it's saturation. And every soil is going to be different. Depending on what the texture of the soil is, every soil is going to be different. Now, the nice thing about this one is we didn't weigh it. We're not adding the water. If you can see, I'm spilling a little bit there. That's okay. I'm not bothered because we're not counting on keeping a certain amount of soil in there. Whatever stays in there is good. We just want to make sure that it's well mixed, and that when we're done, we have what we call a saturated paste. Now you can see that I'm not really saturated down there at the bottom. I've just got a whole bunch of dry stuff. So I might as well go ahead and add quite a bit of water in here just to get it down. Make sure I get some saturation in there. And then this is what I'm going to do. Now I'll come back to this, rather than you watch me do this, I'll come back to this when we've got saturated paste for all 4 of our soils. Okay, so it's been a little while since we started this, but we've made up our saturated paste, and you can see here's D. They're all going to be a little different. Some of them took a lot longer to do, but you can see how it kind of glistens. Now, if I tilt this sideways, I've already done this once and I made a mistake. It'll tilt. You can see it wants to flow, and so it's saturated. Sometimes the clays will tend to want to soak up a little bit of water, so that it's always good to kind of come back and not assume you're done. Here's our second one. It's kind of the same deal. You can see as I shake it, it's glistening. It's very, very wet on the surface. If I tilt it too much and shake it, it's going to pour. Just kind of like when we did our liquid limits, when we did soil consistency. Here's the soil B. This one here, we might be ever so slightly over the top. Sometimes you can't tell, and you get a little bit of freestanding water on the surface. That means you've gone just a bit beyond. But I'd say we have like a 1/4 teaspoon maybe on that one. Same with this. There's a little bit extra on the surface, but it's really not going to make too much of a difference. Is it an error? Not really. Not really. It's not. But you can kind of see that each one of these things looks is pretty well saturated. It looks like just a wet, gooey mass. But it's all glistening. And again, you can see on this one here, perhaps just a little bit too much just liquid on that. If I just pour just a little bit of that out, You can see that's mostly liquid, but you can see how dark that actually is. That's at least 50% soil. So we're pretty close to being accurate on that. So now what we're going to do is I'm going to go ahead and pause this one again in just a second. But the idea is that, and I'll do it now so that you can just watch me demonstrate it later. But what we're going to do is we're going to take this filter paper, because this has got holes in it. This is called a Buchner funnel. This is filter paper. We're going to put this one in like that, and then we are going to put this in this flask. I just forgot the name of it. A vacuum flask. It's called a vacuum flask because it's got a little nozzle right here that's hollow that we can attach an air tube to, which is what we're going to do. We've put this in a rubber stopper so that we're not going to have any air leakage through here. And what we'll do is we'll put the air hose on this. This will actually be attached to a vacuum pump, and it'll actually pull air from the atmosphere through our soil sample, which we're going to place in here. The idea behind the filter paper is, and we'll get the filter paper, just wet it up so that it sticks to the bottom of the Buchner funnel, and then we'll go ahead and extract the material in here. So remember, we've made soil solution, so we're measuring salinity, but what we've done is we've added water with nothing in it, or distilled deionized water, if you will, with nothing in it. We've added it to our soil sample. We've mixed it up to saturation, which means that anything that's in our soil sample is now in that soil solution. It's been gone into solution, and now we're going to extract this. We're going to pull the soil solution out of the sample. In other words, we're going to make a drier sample again, and then we'll have the liquid collect down here, and that liquid will be able to analyze for salinity. So that's the procedure. I'm going to pause now and move all this stuff over to our system to do the extraction, and then we'll pick up there with us doing that. So here we are, ready to do our extraction for soil salinity. You can see the apparatus here. We've got a one-of-a-kind manifold, that's what we're calling it. Basically what it is, this is full of air inside of here, or it's actually full of vacuum inside of here, and then we're able to control the vacuum by opening and closing these valves. We've got a vacuum pump right here. You can see this has got a vacuum. Vacuums always go backwards. They go from right to left to show that you've got a vacuum. And so we're creating a vacuum inside here, so everything is static. Once I open one of these valves, I'm going to actually be pulling air this direction, right? So the air is going to come this direction, but whatever's liquid is not going to go down through here. It's going to actually fall by gravity into this container here. So let's go ahead and start with soil A. This is a brand new run for this particular machine. I don't know how it's going to work, but it hasn't blown up yet. So here's our soil. This is soil A. First thing I'm going to do, as I mentioned before, is I'm going to take these discs. I'm going to put it in there because I really don't want the soil to go all over the place. I'm going to wet it just so it sticks down on the sides. Now, what I'm going to begin to do here is I'm going to turn the vacuum on just a little bit, and I'll show you on the next one what that looks like, because that's kind of actually interesting. So I've got a slight vacuum on this now, so it's actually sucking, and we should be able to see that backing off, and it actually is. So what I want to do now is get this back in if it's settled just a little bit, because it has been sitting for a while. I'm going to mix it up just a little bit again, because we knew it was at saturation before, and then I'm going to, let's see if this will work. We'll get ourselves organized here. I'm going to go ahead and pour this in like this, as much of it as I can. I'm not gonna scrape it clean, but I got some stuff on the bottom there. I'm just gonna get it all in there, a little bit more. And then what I'm gonna do is I'm gonna push it down against the sides. Now this is a trick that I've always used. I push it down against the sides, and since it's saturated, I can actually go like this and get it all saturated like that, and there's not going to be any air bubbles. Now I can fully turn this vacuum on. Now that's got full vacuum, and you can see what's happening already in there is that I'm extracting that liquid pretty quickly. And that's actually the soil solution that we're going to analyze. I'm just going to go ahead and repeat the process. You can see me on this one. Maybe we'll come back to that one in a second. Now you can watch, I'm going to just go ahead and squirt this down again. Okay, we've got all of our samples in there, and we've already even got some cracks forming in this one because this one's really good. We got some good vacuum. This pump can really pull some. So you can see that we've had a crack going on through there. It doesn't matter. I'm going to go ahead and try to repair it. It's not really broken, but I'm just going to try to do it because what'll happen is that you'll end up getting air going down right through that crack, and I'm not really ready for that to do it. I think there might be a little bit more it could extract, and I don't want the air to go down through a crack when it can go right through the soil. And you can see that's actually started a little bit of dripping again as a result of that. So you can come over here. This soil here is a little bit sandier, and one of the things that you have to kind of do is stick your ear in there and watch the gauge, because at some point it will kind of spring a leak in one of these spots, especially when it doesn't have to pull a whole lot of vacuum to get the soil to be extracted out of there. Soil C and soil D tend to have more clay in them, and you can kind of see that the surface on that is a little slicker. And it's going to take a little bit of time to really get those all pulled out, or for us to get a good sample. You can see really nothing's even happened on this one yet. We don't even have one drip coming through there because you've really got to create quite a vacuum to actually get that soil, the soil solution to actually pull through the whole soil sample there. So we'll let these run for a little while, and then we'll come back and we'll pull them off, and then we'll show you how to analyze them. Okay, so we're still on lab 9, and we're switching back and forth. So I'm going to run this whole lab, exercise 9, as one not so big, but one complete video. So this is part 2. We haven't finished part 1 yet. We're just going to go to part 2, and part 2 is pH. So again, this is the way the lab would do it. There's other ways to do pH. You can use pH paper. You can use colorimetric, which is actually what I do in the field, where you take a little pinch of soil and you put it in a spot plate, and you add different chemicals to it, and then you compare it to colors on a chart. And it's not inaccurate, but it's no substitute for using a pH meter, which is what we have over here. So we have a combination. Well, it's a multimeter actually, but we've got a sensor for pH, or an electrode for pH, and another one for salinity right here. So we'll run that in just a second. This is the same machine that we'll use to get our salinity off of the manifold in the first part of this particular lab. But for us to do pH, the way we do pH in this part of the country is what we call 1-to-1 soil to water. In other parts of the country, because of their soil pH, they're going to use a different method. But here we're in California, we're going to use a 1-to-1. So what I found is that what seems to work best, so that you have enough depth for the electrode to work, is I weigh out 40 grams of soil, and if you remember what is our analog for milliliters for 40 grams, it would be 1 gram equals 1 milliliter, right, when we use a water basis. So we're going to use 40 grams of soil, and then we're going to use 40 mils of water. That would make us a 1-to-1. We're going to mix those together, and we'll let them stand for 5 minutes or so, and then we'll be able to measure those with the pH. So let's go ahead and do that right now. I'll just start with the same soils that we use for the salinity, and let me get myself going here. Go ahead and tear a weighing plate, and I'll start with A, and I'm just going to go ahead and fill it up with 40 grams of soil. I'll show you one of these, and then you'll know what the rest of them look like, and I'll just fill them up. You don't have to watch that. 39.6, 39.8, 39.40. What's nice is when you have only one decimal point, it's pretty easy to get 40. So I'll just go ahead and just take one of these, and I can just use this. It'll take just a second, but I'll go ahead and fill this up with my distilled water to 40 milliliters of water there. Okay, so I'm at 40 mils. So what I'll do is I'll go ahead and pour this in the one I've labeled A, and pour my 40 mils of water in there. All right, so there's my 1-to-1. Now I'll just go ahead and mix it up. You want to let it sit for a little while because you're trying to put everything into solution, and you're not measuring the soil pH. You are, but you're doing it indirectly. You're doing it by measuring what's gone into solution, which is why you need to use the DI or distilled water, so that you don't already have some sort of stuff in it that's going to be messed up by you actually trying to read what's already in the water plus what's already in the soil. So that's why we start out with a 7 pH, if you will, a water, and then when we get to the soil, we're really just measuring what's actually dissolved, gone into solution from the soil into the water. But for that to happen, it's not an instantaneous process. So you wait about 5 minutes or so. Some people wait a little bit longer, but minimum of 5 minutes, and you're not going to get a better reading if you wait for, like, say 2 weeks. But 5 minutes is usually good enough for all that to go into solution, and then you're actually having an accurate reading. So we'll go ahead and finish the B, C, and D soils, and then we'll come back, and in 5 minutes or so we'll be ready to measure those. We'll probably be ready to go ahead and measure our salinity as well on those. So we'll go ahead and put a pause here, and then we'll come back when we're all done. Okay, we've gone ahead and waited a little bit more than 5 minutes on these. We've got our pH samples ready to go. Here's the machine. It's facing towards the camera so you guys can kind of see it. So I'll show you the procedure. There's 2 electrodes here. This is the one that we use for pH. You can see it's sitting in a bottle. This is intentional because we don't want this electrode to dry out. So this is like, what do they call this one? It's potassium chloride is what it's actually sitting in. So what we do is try not to drop it, of course. And you can just refill the bottle. It doesn't really matter if it gets dirty or anything. But it's important to go ahead and slide this up. This is all new, so everything's a little tight. Slide that up like that, and then we have our distilled water here. That has a pH to it. So what we're going to do is we're going to go ahead and rinse this off with DI water here like that. Just rinse it off, and then we'll go like this. Okay, so I'm going to go ahead and start with A, and I've got my trusty secretary off camera here, who's going to go ahead and record the values for these different ones, so you guys don't have to remember it, but you can kind of see. So I'm just going to gently mix it up. Try very, very carefully stir it. It's really not always advised to do that, but if you're careful, you can do it. Just do not jam your electrode on the bottom. So what's happening over here is I'm watching this screen right here. This is the pH side, and this is temperature compensated. When I get the little happy face there, it means that it's actually ready to go. It will fluctuate. Right now it's fluctuating, I'm stirring it. So the pH here is 7.78, which is really 7.8. So that's A. Now, I'm finished with A. So what I'm going to do now is I'm going to go ahead and rinse this off again between samples. I'm going to rinse it off, make sure that electrode is clean. And some people actually take the added step of not just shaking it, but then they take a little laboratory tissue and they wipe it down. I think if I shake it like that, I'm pretty good. I'm not too concerned. So I'm now ready to do the next sample, which is B. Very carefully stir this up. This is a sandier soil, and it sometimes takes a little bit of time for these things to find their spot. So we'll just wait and just gently stir it, making sure we don't grind the bottom of the electrode down. This is 7.996 is what it jumped out as. So 7.96, that's actually closer to 8. We'll just round it to a pH of 8 on this. Okay. So that's, rinse it out again, shake it off. Let's go to soil C. This one's 7.65. We'll just let that one go 7.6. Now, I'm not sure where these soils have come from. Some of them I think I know. But this last one, I'm not sure where that came from. So we'll try. I mean, I'm surprised that my numbers are as low as they are. Let's just see what we get on this last one. This is D. Ah, now we get something that's very interesting. I kind of suspected this might be the case. Okay, so soil D has a pH of 10.28 or 10.3, if you will. 10.28. Now, that's pretty high for a soil. It's not unheard of course, but that's pretty high. I think we're going to find that soil is going to have some interesting salt issues as well. So I'll make sure that this data is all available to you. I might actually give you some more data to sort of play with, solve some problems. But you actually saw some variety through here, especially with that last soil D. So I'm going to go ahead and put a pause on this one, and then we're going to go get it ready to do our extraction, and we're going to read the salinity. I'll come back. We'll switch electrodes. We'll use a conductivity electrode, and we'll come back and we'll do the reading on the other side of this. Okay, so we're going to turn this salinity extraction off. The way to do that is quite simply unplug it. Now, before we do anything else, I want to go ahead and let the pressure off. So I just let one of those things open up, and once it finishes cooling off, we'll be able to take these things off. Now, it's kind of interesting if you look in here. Okay, we're empty. So now I can actually pull this off. So hopefully I don't break this, but there we go. So see how I made it nice and clean because I put that filter paper down, and now we've got this nice like a cake almost of soil here, and that's just the extraction, just sucking it all in. So what we're going to do now is I'm going to disconnect this and make sure I keep them in the same beakers. I'm just going to go ahead and pour this out. This is the stuff that we're going to analyze. That's our soil solution that we extracted. Soil A. Interesting color, right? Soil B. This was a sandier one. This will probably come out quite easy. There we go, soil B. And all right, soil B. Do the same with soil C. I'll save you the banging for that one. There's soil C. And finally, you can see we could have extracted more out of this, but it would have taken us some time. This is still pretty sticky and pretty wet. Now, I'm going to put these here just a second and grab the other ones. So you can actually see there that there's varying amounts there. So it was all saturated, but even though our soil textures held different amounts, we were able to, with the vacuum is not like the world's most powerful vacuum. So it will take a while for us to get these soils completely dry, and we waited quite a while we did other things for that to happen. I stopped where I thought we had enough extract for us to analyze this, and that's what we'll do with that one there. But again, some of these textures are going to release their water, the soil solution, much more rapidly than other textures. Now you do remember that we just had a pH of 10 on soil D, so this is the same soil D, and I'm going to suspect that we might I don't know because I've not done it before, but I'm going to suspect that we might also find that soil D has some interesting salinity things going on as well. So I'm going to go ahead and pause quickly. I'll get the stuff all set up. I'm going to move these beakers over to the apparatus, the machine, the meter, and then we'll pick up from there in just a sec. Okay, so we're back over here at the meter, at the conductivity meter now. One thing I might want to say is that I put the soil sample D into a smaller container so that that probe will be able to immerse, and hopefully that's deep enough. If it doesn't work, then I'll put it into something a little bit smaller so I have a little bit more volume and measure that. So here we go. So A, B, C, and D. I'll go ahead and move that out of the way. Okay, let me just explain. So we're measuring salinity by way of electrical conductivity. So what this machine is actually measuring is measuring the conductivity. It's passing electrical current through this electrode here, and through the solution, and then back into the electrode, and it's taking a reading of how much electricity is conducted as a measure of resistance. Pardon me for my less than stellar explanation of electricity and resistance. But that's kind of sort of what's happening through there. So probe, this is a probe on this one. Same kind of thing, this thing's been set up. One of the things I didn't mention last time, it's temperature compensated, so that would be, we'd have to do that formula calculation ourselves if the machines didn't have that built in. So no matter what the temperature is, and it feels pretty good in here right now, this thing takes into account, it's got a little temperature sensor in there, and it'll actually read that. So let me go ahead and get this thing organized, and we'll start in here, and I'll show you what we get. So here again, soil A, got no idea what to expect. Okay, so this is about 548 micro Siemens. What this machine does, it's got several different ranges. So it kind of runs through its different ranges, and it says, oh, nothing on that one, oh, nothing on that one, and it goes down to the smallest range. So 535 sounds like it's a lot, but it's a micro Siemens. It's not a high value. I'm going to do the same thing. I'm going to go ahead and rinse this probe off again, just like I did with the pH probe. Shake it off a little bit, and then itch my nose, and then we're going to go ahead, make sure we're good there, and we'll start with B. Okay, so this one's come up with at 1437, 1,437 micro Siemens. So the Siemens is a standard unit. You'll also see other terms in the literature like millimoles, like mmHOS, millimos. You can convert electrical units just like anything. It's just like you can convert inches into centimeters or meters. You can do the same thing with most of these units. But Siemens is the common unit, and it's usually it usually has a length attached to it as well, centimeters, meters, something like that. We'll convert everything into a standard so that you can understand what the different readings mean. So if we're talking micro Siemens or deci Siemens or milli Siemens, you'll know what they all are. So I'll go ahead and make that conversion for you, show you what the reading is, and then show you how we convert those into something that you can actually compare apples to apples, if you will. Okay, so here's C. Let's see what C says. Okay, so this is 19.59 millisiemens. Okay, we had micro Siemens before. This is the next unit up. So this is millisiemens, 16.59, I think I said.
1959.
Say it again.
1959.
19.59. Thank you. See, I'm glad I have people helping me out here. 19.59 millisiemens. Okay, so here we have D. You can see I'm just going to shake that off. I'm going to tilt this so I get enough to get that probe in there. It looks like I will. Okay, this is similar. This is 15.42 millisiemens. So we had two that were micros and two that were millis. So I'll go ahead and put these all into the same units so that you're able to know what I'm talking about, because even for me it gets confusing. I got to go back and remind myself which one is which. So there you go. That's how we do our salinity. We go ahead and rinse this thing off again. Make sure we got that probe nice and clean. And then I can go ahead, I should, if I was some tissue, wipe it off like that. I don't have that. I'll just shake it and then just go ahead and put that cap back on the bottom. Keep that thing protected and you're good to go on that one. So now, I'm thinking that I might keep some of these solutions, and there's a further test that I'm not going to be able to video right now. I might be able to video it later, but I'm going to keep these solutions, and there's actually something else that we can actually do with these. So what we've actually measured right now is salinity, and that goes hand in hand with the pH. And by how high the pH goes and by the numbers of salinity, we know there's salts, but we don't really know how much of those salts are actually going to be sodium. So there's a further test that we can take this extract right through here, and I'm not set up to do it today, but I can keep my extract. It's not going to go bad, and I can actually run this other test, and I think I'll probably do that. I'll put these in some bottles that I can cap. I think I'll probably do that, and I'll give you those numbers. If I can, I'll take a video of me doing that as well. It might be more challenging, but I'll try to do that. But if nothing else, you know that I can take this and further analyze it, not just for salinity, but for specific salts. So I can look at the total soluble salts. I can look at how much of those salts are calcium. I can look at how much of those salts are sodium. And that might be instructive for us to know that, because soil behavior and then how we're going to feed our soils, nutrients, all that kind of stuff is going to make a big difference based on what the actual composition of salts in there is. Okay, so one more thing to do. We'll put a pause here and get set up for the last one. One more thing to do in lab exercise 9, and then we're done. Okay, so this is part 3 of lab exercise 9, and this is called determining calcium carbonate equivalent, or what's abbreviated as CCE. So in this particular lab, this is not something that's done very commonly. You're not going to see people do this. It's an automated procedure in some cases, but originally it was all done by hand, just like we're going to do it. Now the reason for doing this is that in many parts of the country, not California necessarily, but many parts of the country, they want to know they have acid soils. And we don't have that problem in California or in the West. We don't have acid soils. We have soils that are the opposite of acid. But in many parts of the country, the pH of the soils is pretty low, and they want to actually raise the pH. And how they do that is that they use a liming agent, which we've talked about, or we will talk about in class. But every liming agent has a different amount of calcium carbonate. And calcium carbonate is the ingredient, if you will, in these different liming agents, or these different materials that we use to raise the pH in soils. Has a different amount. So every substance has a different amount. But pure calcium carbonate, we know what that is. So we give that a unit of, say, 1, and then everything else is measured to that unit of pure calcium carbonate. So we've got a couple different soils here. I only have one set up. Again, this is soil D. This is the one that had the high pH, but I've already checked it out. I know it's got lots of calcium carbonate in it. I have no idea how much it's got. But what we'll do in this procedure, and like I said, I've got it set up here because this is getting to be a longer video, is that we weigh a very, very small amount, 0.33 grams. That's 1/3 of a gram of soil. And we're going to put it into this large syringe here. The syringe has got to be at least a 50 ml syringe. This is a 60 ml syringe. I've lubricated this rubber plunger so that it slides relatively easy. That's going to be kind of important. You'll see that in a second. So in this small syringe here, which has got a little rubber sleeve on it, I've got dilute 10% hydrochloric acid. So I've taken concentrated hydrochloric acid, and I've gone and oops, I didn't mean to do that. I've gone and diluted it down to what we call 10%. So this stuff is not really, it'll eat through your clothes, but it's not going to, if I touch it, I'm not going to go screaming or anything like that. It's totally fine. We use it all the time out in the field, put in little bottles and whatever. I have had some clothes ruined with it, but no big deal. But in here, I've got 5 milliliters of this 10% hydrochloric acid. So what that hydrochloric acid is going to do, it's going to react with the calcium carbonate in my sample, and it's going to generate carbon dioxide. That carbon dioxide I'm going to be able to read as a volume inside this syringe here. So the first thing I got to do is I got to put my sample in here. So I take this off and I put my finger underneath there because I really don't want all this stuff going out the end. And then I very carefully just pour my pre-measured sample, and you can see this is really not very much. You can kind of, I don't know if you can see in there, but there's really not very much in there. It's just a little tiny amount. Oops. So there it is. There it sits in there. Just a little bit of stuff. That's probably not, and now what I'm going to do is I'm going to again hold my finger over the end there. I'm going to stick this syringe in the bottom. Now I'm just going to tilt my finger like that, let go of it, tap the stuff down here, and now I'm going to push this all the way up. I don't want to blow any of my soil sample out the top. Put that all the way up like that until it's flush against there. I can tap it a little bit if I want. And now you can see that my sample is on the end of that syringe. Okay, so now what I want to do is I want to attach these two together with this little rubber sleeve. And so now I'm going to, now that they're attached, I'm going to inject this hydrochloric acid, the 5 mils of hydrochloric acid, into my sample. So you can see I'm not touching it, I'm just injecting it in there. And you can see the plunger of the larger syringe is actually going backwards. Now, if you can zoom in on that, you might see that there's actually there's a lot of effervescence going on inside there. So it's wanting to push it back the other direction. So I'm just going to go ahead now. What I'm going to do is I'm going to actually pull this syringe down, and I'll have to fiddle with this a little bit to get a nice volume of air going on there. But so you can see what's happening here. I'm going to wait for it to finish bubbling. And sometimes you don't want to take the chance that it's just the weight's going to pull it apart. But this is a sealed system now. There's no way the air can go out. So all the carbon dioxide, which is a gas that's been produced, is actually causing this interface. So what I'm going to try to do is I'll shake it up a little bit, make sure that there's no more calcium carbonate in there. Now, what the reason for the 5 mils of sample and only the 1/3 of a gram of sample in there, or 5 mils of hydrochloric acid, is because you can generate, some of these little tiny samples can generate huge amounts of gas, huge amounts of carbon dioxide. So it's all finished bubbling. So I want to see exactly how am I going to measure that. So you can kind of see that top one, I'm pulling it down, and I'm going to have to help it a little bit to get going. But you can see I'm going to push a little pressure on the bottom one there, and you can see it wants to go up. So I'll push it back down. It still wants to go up. So I'm pushing against the gas right there. That means that this is full of gas in that whole volume. So I'm going to pull the bottom one down a little bit and push that. Try to find an interface. If I get a good syringe, then it'll actually work back and forth. So what I'm going to try to do is just find the spot where it's not going to go up anymore. It's right about there. It's just barely breaking the seal. So I know that if I push this bottom one up anymore, I'm going to actually create quite a bit more stuff. So I'm going to push that back one up there. But you can kind of see where we are right now. So my sample was 0.33, which is negligible in this large syringe. But you can see I put 5 mils in, so I haven't lost my 5 milliliters of hydrochloric acid. It's still there. But the difference here is that I've generated quite a huge amount of volume. So what I really want to do is I just want to measure how much volume I've actually created. So if I go down to the top of the liquid through there, that's not everything from the top of my syringe, because I've pushed it down. This syringe doesn't have anything in it. I've pushed down that whole amount of carbon dioxide gas that's been created. All I got to do is just measure from 0 here, which is right there, all the way down to, and this one looks like it's just about it. Well, I'd have to read that at 20. So I've actually created 20 milliliters or 20 cubic centimeters, if you want the same thing, of carbon dioxide gas in that little tiny sample. So the next thing I do, so I write that number down, 20 milliliters or 20 cc's, and now this is what you're going to get. So what you're going to actually do is you're going to actually take this little sheet. This is called a nomograph, and I'll actually give this to you. All you got to do is just kind of look at it. It'll be the only thing you ever see like this. And then you're going to actually, based on your elevation, based on the numbers I give you, you'll be able to determine for soil D and whatever other samples I might choose to give you, what your calcium carbonate equivalent is. So what % calcium carbonate you actually have in this sample. So that would actually then tell you how much, based on your results of your pH results, how low your soil or how acidic your soil was, how much of that particular product you would have to actually use to raise your pH to a certain amount. So this is basically saying how effective is this particular material going to be based on how much calcium carbonate it has to be able to raise your soil from whatever pH it is to whatever pH that you want to get to. So that's the end of lab exercise 9. See you for the next one.